Thank you very much, Amir. Thanks for inviting me to talk about our research and others. What I want to do today is basically to go a step beyond the methods we heard about, in the sense, what can we do with all these nice technology? And in my talk, I will wrap up a few of the conceptual topics we heard this morning, and then I will focus more on what these new techniques provide as opportunities to study representational content in the human brain. Since I think that the high-field fMRI, with this what we call often mesoscopic scale level, allows us to see structures which we heard about. We heard a lot about layers and a bit about columns. I will focus on both, but mainly on columnar resolution, and will show you why I think that we have here kind of an impact on how the future potentially of cognitive neuroscience and computational neuroscience will be. And it is important that we kind of bring these methods to cognitive neuroscientists so that it's really used more, because I think there is such a high potential that is not yet exploited enough in the field. That's at least my personal opinion. So what I will do is I will focus more on results. Just a little bit on methods, mainly on results. What has been shown already with mesoscopic fMRI, that means columnar and laminar fMRI. And then I will even go a bit further and show you some new studies, some very recent studies, which point in the direction that we can even investigate in a new way conscious experiences using these kind of ultra-high field MRI studies. And I will, throughout my talk, point to some implications of this new field for cognitive and computational neuroscience. So first, I want to stress that if we have methods which more deeply invade into higher resolution, we can say that we can better relate different scales of the brain, or activity in the brain, to each other, and we can use maybe more finer spatial scales and what's happening there to explain what's happening on a lower spatial scale, which gives us much more predictive power and kind of explanatory power to understand phenomena at a higher level. And this kind of, on the one hand, reductionistic but also emergent phenomena perspective means that if we relate the different scales, then we have not only the feeling and the reward of a deeper understanding how higher level concepts are grounded in the lower scales, but we can also reconstruct things happening at a higher scale from knowledge at the lower scale if these reductionistic explanations are valid. And I will follow a bit this approach here. And to kind of stress a bit what this kind of high resolution, high field fMRI brings to the field, I show here a slide which shows that and compares a little bit conceptually what the gain is from traditional fMRI to ultra-high field fMRI. For example, here is the resolution scale of, say, 2 millimeter or higher, which we obtain by conventional MRI, 3 Tesla, both for structural scanning and functional scanning. Unfortunately, my mouse doesn't work. Is there a way to get the mouse work? If not, probably not. No. I will try this out. I will point to things. You see here what we can achieve with conventional MRI at a macroscopic scale. And now if we increase the resolution in the sub-millimeter regime, people always think, okay, then you get your voxels a bit more higher resolved, and maybe if you do not use bigger voxels, a bit better signal to noise eventually. But the point is here that we get a qualitative change, not a quantitative change. Because with a slightly increased spatial resolution, which is, as we heard this morning, very effortful to get from the physics and analysis point of view, but if we get this kind of from a few millimeters down to sub-millimeter spatial scale, we see new things. We have new windows on the brain, because we can reveal lamina, as we have just heard in the last talks, but also columnar structures, as we heard in a talk from Amir and others. So that's very important, that we kind of not only get more of the same, but we get qualitatively new structures to image. And these structures can help us also to link fMRI better to animal research. Because, like Renzo showed earlier, we have here a bridge between lower scale methods, like electrophysiological methods, optical imaging, which kind of reach up to kind of resolutions of columnar and laminar scale. And that's now where we can shake hands from the animal research to the human research and relate things better to each other. And I think that's another advantage of this field. So, but why is this all possible? We heard this before, so I will be very brief on some concepts. But I want to just point out that the columnar concept was often criticized, especially from Horton and colleagues, and not that it doesn't exist. Of course, you observe them empirically. The point is whether evolutionary columns have a value. There are species without column who have also very good, say, vision or haptic sense. So has the column a certain deeper advantage of organization? That's an open question still, but I have to say that for fMRI, I don't care. So what we for high-resolution fMRI just want is that neurons with similar response profiles cluster. Since if they cluster, we have a chance to see them with our still lousy resolution of, say, 800 millimeter isovoxels, right? So to achieve that, what we really want is that neurons with similar response profiles cluster and don't spread randomly within, say, a tiny piece of the visual field in V1. If then the different cells, like here in this cartoon on the upper right side, from different orientation features would not cluster in columns, but would kind of be randomly spread around, even the increased resolution we get on 7T and higher will not be enough to resolve the features. And therefore, we are just lucky, whether it has a meaning or not, that neurons cluster, that we find that we have this, here textbook-like, but it's called much more noisy, but we have this kind of columnar-like organizations where, as Amir showed nicely with the electrode recordings of Goebel and Wiesel and others, that if you penetrate vertically, you find that the responses of the neurons is roughly similar across depths, versus it changes more horizontally in the cortex. So, and if we use these findings and apply them now to ultra-high field, then of course other people and also our group and many others have shown that's indeed feasible. For example, here is a pioneering study from Isa Yacoub and the group of Camille Goebel, which showed that if you combine spin echo EPI, which you heard also this morning, and 7 Tesla, you can, with a single slice, I come to this in a moment, you can map in V1 in the human brain orientation columns and ocular dominance columns. So the black lines demarcate ocular dominance stripes, and the colors show you the orientation features mapped out similarly as you find with optical imaging in the monkey. So we could also say, if we are lucky, we get something like a non-invasive optical imaging technology, which even can penetrate in depths. So it's a very principally cool technology. The only problem is it's full of noise, as we heard this morning, and we have to be very careful. So why this study was successful to resolve columns? Because we heard also that you need a high resolution to resolve quite small columnar structures. Here, the in-plane resolution was 500 micron, but it was only one slice, so the depths of the slice, which you see on the right side, this red line, this was the slab, and it has a thickness of 3 millimeter in depth. And the subjects selected were those in this study which had a straight calcarine sulcus. So otherwise, you would, if it would be curved, you couldn't image it with one slice. Right? So there are a lot of prerequisites here, and the depth is actually good for pooling, because you pool along the depths, because the columns should go in V1 vertically down. So a thick slice is good, but you need tangentially a high resolution to resolve the different orientation features, and that's precisely what the study has done. But when we started our efforts in our center in Maastricht to image columnar structures kind of everywhere in the brain, in principle, we faced the problem of curvature. And so we like many others, like you heard a nice talk of Polymeni earlier, we always usually visualize our data on flat maps. But single flat maps are no longer sufficient. We have to model the cortex as a three-dimensional structure, because we have no depths in the cortex. We have tangentially, potentially columns, and we have in depths the different laminae, which help us to do directional connectivity and other interesting topics. And you have seen that before. I will not go into much detail, but I want to stress that for columnar imaging, you cannot pool across a horizontal space, and that's a severe problem, why we develop specific sampling tools which are not using the normal triangular meshes of free server or brain vulture, because they are too irregular. So we develop a new kind of reconstructive flat maps in depth space, We call them two-dimensional Euclidean grids, where everything is known and you can do this exactly as you need. So it's a reconstruction of a new kind of mesh to be better suited for these local analyses which we do. And we also use the Bock principle and the Wiener idea about how to correct sampling of layers with the equivolume technology. I have not to describe this. Jonathan Polymeni just did that perfectly well. I just want to highlight that if we then put these grids in the cortex, we can sample both along, so to say, the columnar direction, as well as tangentially in a regular space along our grids, where we hope to find columnar structures. We can even, because if we use a version of this approach where the regular grids are kind of correspondingly put on top of each other, we can even make volumes out of this. So basically we flatten the 3D cortex for small regions of the brain, not the whole brain, but then we get regular volumes back, where now you have two cortical axes and one depth axis, and you can use all tools for analysis which you have developed for volumetric space too. So coming back to the difficulty of sampling, for columnar study, I want to stress that for laminar approaches, we have very smart approaches from the group of David Norris and others, where the horizontal sampling can be done if you are interested in laminar profiles over a large extent. And Jonathan Polymeni just showed that the larger this extent is, the better and the more precise will be our laminar sampling. So that is one approach. I call them the horizontal or laminar pooling approach. The other we saw in the Isa Yacoub study, we have also depth sampling. Here we had horizontally 500 micron, but 3 millimeter depth in this one slice, and so we pool, so to say, vertically along a column. But for the work I want to present today, for most of the work I present today, it's important to resolve both at the same time. So we face the issue we cannot pool. That means we try to interpret and analyze the voxels really independently and individually, and use mainly univariate mapping. Of course, all three, that's why they're here in my highlights now of results, all three approaches are used. But I just want to stress that the studies I will talk a bit more about use the more challenging approach, because I think that brings us really also to a new insights. So first, I will show you a few highlights, what has been done in our and other labs about mainly columnar mapping. Then I will go to more laminar mapping and select only two studies, which I found very exciting. And then I come to finally to our recent application for consciousness research. So first, when we started to do fMRI in the 90s, this was a highlight of our work. We used a vision scanner from Siemens, 1.5 Tesla, and got beautiful mapping of MT by contrasting motion versus static. But actually, we also did on 3T later, but this is the original 1.5 images here. But what I want to say here is that important point was that at that time, no one asked the question, can we map directions of motion? Because that would require that you have a resolution that you not just get MT as a blob, as an area, active for motion and less active for static, but that you really see subcompartments inside that area for different directions of motion. And MT is not V1, it's not straight, it's on curved cortex. So we need also the new analysis tools Jonathan has shown and similar ones we have developed. Otherwise, you could not analyze that. So the question has now turned with high field imaging in the question, can we map the features in human MT with 7 Tesla, and this is direction of motion. So we tried that, and if we used motion directions in different 8 different directions, using actually the model which Scheimhoff has presented this morning. We used it actually for our study some years ago, a very early version. We re-implemented that, and we predicted that for direction of motion, the resolution we had at that time, 0.8 millimeter isovoxels, is not good enough to resolve the columnar structure using the Scheimhoff model, which we saw earlier. So what we did finally, and Scheimhoff was contributing to that, and Amir. So what we did at the end, we pooled opposing directions of motion, because that gives you bigger assumed or expected clusters, bigger columns. Because from the animal literature from Albright and colleagues, in this very idealized model on the right side, you see that opposing directions of motion neighbor each other, so that you get together. If you just look for axis of motion, it means motion like here and motion like here is one feature, motion like here and motion like here is another feature. You get bigger clusters if you look for what we call axis of motion. So we did that, and you see here the result. You see on the left upper side this little piece of colored cortex. These are the depth layers we put in MT, in the human MT, mapped MT, localized MT. See this a bit zoomed in on the right side. And the lower side, you see two depth planes. With this grid technology here, they are just flat grids. You can just visualize them as is. And you see now the different axis of motion colors. So these are 4 different axis of motion mapped. So these were the first maps, an extrastriate cortex of columnar organization in the human brain. And that was very encouraging to move on to other pieces of the cortex. Also important is that the tuning curves we found for the voxels sampled for different axes of motion looked unimodal, and the full width effect maximum was similar to what we expected from the monkey research. So we were kind of succeeding in this first approach to map another area, not V1, the features inside. Then we moved on, for example, to disparity, just to show. And actually, I come to disparity in V2 in a second. But first, I want to stress here we looked for disparity in MT, because MT has not just one map. It's not just direction of motion. It has also maps for disparity, for depths. So you not only see with MT that something moves a car from right to left, but you also see with MT in parentheses how far the car is away, the depth, the distance of the car, right? And so we mapped in the same area and the same subjects disparity. Disparity means you look with 2 eyes and you basically get 2 images on the scene, and from this your brain can calculate some distance, some depths in the scene. You know, and you see this here, another example, and we basically followed again work on animal literature, and I told you we can link animal to human research, so we followed clearly a paper from De Angelis and Newsom, and basically redid this now with high field imaging in the human. And we also expected, like on the right side, these kind of colored green, yellow, red zones are a fixation plane, and more far away and more close should be larger columns detectable in MT. And actually we found these, exactly. We found a very, very nice columnar organization for depths now in the same region of cortex that we found in the same subject, different maps for direction of motion or axis of motion. And that's, of course, striking. We are still working. The MT study is of course, is published, but this not yet, because we still try to relate both the direction of motion and the disparity maps to each other in the same area, which is a fantastic phenomenon, that in the same area you have kind of somehow interspersed or somehow mixed maps of different feature spaces. They're, of course, related because you want to know where the motion is in depth. But nonetheless, you get different feature maps if you do it. And this might require some more modeling, which we're currently doing, to understand this better. So now I want to go to go back to B2. Actually, the study here by Nasr, Polimeni, and Tutel was a very nice study, which again linked human fMRI to animal research. So they started with the shown, and actually Amir showed us some of this before, of the cytochrome oxidase histological staining. And actually, the thin and the thick stripes have different feature specificity, which has been found. Namely, the thin stripes are for color responses, and the thick stripes for, again, binocular disparity at an earlier stage in the brain, in V2 here. And indeed, these authors found that you find, right after V1, stripes emanating towards V3, which kind of have a higher response to colors stimuli. And even more exciting, if you look at the same subjects across V2, V3, for color and disparity, you see that they are interdigitated as expected from the animal research. That means color stripes and the so-called thin stripes in the monkey, and the disparity stripes, the thick stripes, are indeed interdigitated, as you see nicely on the right side. I have no mouse. So the right column shows you outlines of disparity and yellow-filled stripes from color, and you see that they're nicely interdigitated, which is what you expect from the animal literature. So we can map the cortex various feature spaces. Here's another one in the auditory cortex. So work from DiMartino, Formisano, and colleagues in our group in Maastricht. You see here that we can image and map with grids over the Herschel gyrus on the left upper side. You see grids on the auditory cortex, and we can find indeed that these kind of tonotopic maps, which kind of respond to different sound frequencies, and the color shows you low to high frequencies, is nicely mapped there. And you see on the right side, indeed, that they penetrate vertically. So the frequencies in A1, in the primary auditory cortex, are nicely organized in a columnar fashion. Whereas if you go around in the belt regions, you do not find very nice columnar organization. So that shows also that we can now use fMRI to go also in areas where we might not have animal data available, or the human areas are different than in the animal. And then we can now start, if we now trust the methods and trust this kind of technology, we can also go and reveal new features in new areas, and that's very exciting in my opinion. Another study which gives us some warnings, actually we recently published, where we compared in the same subjects increasing spatial resolution. Actually, there was a lower one than the 1.5 by 1.5 by 1.5, and this one is the middle one, and we have a high resolution one, 1.1 isovoxels. And what we found here was a bit scary, namely that if you publish, for example, how voxels are distributed for different features, in that case, we have here stimuli of visual and auditory input, and there can be concurrent and not, but I will spare this. So you have visual, purely auditory input, and combined input, visual auditory, multisensory input. And we measure here in STS, superior temporal sulcus, where it's known that these columns for different modalities come together. So if we look at low resolution data on the left side, you would come to a completely different picture of the proportion of columns, or say topographic space for different features. You would say most is auditory, much more. Visual is almost negligible. But if you increase higher resolution, you find that visual bumps up and auditory goes down. And the reason is that if your voxels get finer and finer, then if you have a big voxel and auditory dominates, and you have, say, a third of green and 2/3 of red, you will classify and give a label to this voxel. It dominates its response auditory, so you label it as auditory. So that means we need to come closer to the true expected organization. We need to do the modeling, as we heard from Trimov, also in such areas to predict what is the right resolution to understand these kind of columnar organizations. And when we pushed it higher and higher, we found that we got to a more steady state, which is also distribution expected again from animal experiments. But you see on the right upper, just look at the right upper graph and compare to the left, the sensitivity and specificity boosts a lot, because if you now sample with higher resolution from a column which is really purely visual or auditory, you get basically only response for that modality and not for the other at all, which you never see if you have 3 millimeter or 2 millimeter voxels. And even more, we for the first time found what the multisensory community calls super additivity. We find that multisensory voxels respond not at all to individual visual auditory, but massively if the two are combined in a concurrent way, I should say. So if a dog barks, it's higher than if a dog meows, right? The semantic concurrence plays also a role. So now at the end of my talk, I want to tell you a little bit where I think that this field is heading to, namely to bring also something more and more to the cognitive psychology, cognitive neuroscience people. And I think that some of this we have already heard in the talks. If we look at the directional connectivity from Renzo, from Jonathan, from many people before. But I want to go also after showing you two other studies not shown up to now, on laminar fMRI. I will then go to columnar fMRI and how we can study consciousness. So first on the laminar fMRI, this I will skip because of time. Here's again a nice study from David Norris' group. Actually, Floris de Lange is the senior author here, and Koch the first author. Very nice study, which I found extremely elegant, where they studied top-down effects and the laminar resolution, when you make a Canisa Pac-Man-like figures, rotate like on the left upper side, rotate them in a way that you get an illusory figure. You see this kind of triangle in the middle of these inducer Pac-Mans. And in the right side, when you just rotate them differently, you do not see this inducer, right? And they find that if you reconstruct the effects using PRF modeling, there where the illusory, if you compare the constructed illusory triangle versus not constructed, so A versus B, you find that in the empty space, so to say, you get a strong top-down influence, kind of indicating that's the space in V1 where this kind of illusory triangle is constructed. Right? There are many details in that study. I have not the time to go all into these, but this is one very elegant study demonstrating how you can use now laminar fMRI to study something very cognitive, predictive coding, top-down modulation, expectancy, attention. All these phenomena become now in the reach of research for cognitive neuroscience. I will just show a point of the upper figure because here we sees very nicely, and that's a little bit for me puzzling. Their effects were in the deep layers. See, the green curve is higher for deep layers than for the control stimulus, whereas in the study I show next, we find in the study that upper layers play a more important role. So we have to find out why different researchers find very nice laminar top-down effects, but in sometimes in upper, sometimes in lower layers. And this is the study I want to go to from Lars Mukli in Glasgow. And what he did was, again, also to look for top-down effects, contextual effects, in area V1. And what he did is, in an older study, he used MBPA-like techniques, decoding techniques, to classify from the activity in V1 what a person sees on the screen, which has been done hundreds of times. But on the right side, this is the cool stuff, he occluded a quadrant. You see in the test column on the right side, you see this lower white square. And that shows that in this experiment, there was no stimulus to the right lower quadrant. And nonetheless, he used the voxels in V1, in the left upper quadrant in the visual cortex V1, which is not stimulated, but he still could decode what the person sees on the screen. So how is this possible? So the idea is that if you see an occluded image, it runs up the visual hierarchy. It is completed to some extent. For example, in the car scene, you know at the right side must be car features, right? So you kind of complete that. Then you project this down into V1, including the region which is not stimulated. And then in this region, you can then from the activity, top-down activity, still classify what image a person sees. And this was not a laminar study, so we've joined forces and combined and did this work using laminar resolved fMRI, and we found that if you show the full pictures in this kind of quadrant, the voxels, you see the red line on the right side, that the middle layers, which are in the middle of the curve, are decoding what people see close to 100%, and the upper and lower layers are also very good if you have the full stimulus. But if you look at the green line, that's the line which corresponds to the lower right green stimulus, and only voxels are used which are not bottom-up stimulated. And you see that at the very right side, the accuracy goes up sharply, and these are the superficial, the upper layers, right? The superficial layers. So these are able to still tell you, although there is no bottom-up input, what is currently seen. And since we now have a laminar resolution, we can say that the information is contained for MBPA in the upper layers. That's what we can add to the previous non-laminar study to that. And if we look at other subjects, we have a consistent picture. So always the upper layers basically contain this information. So now finally I want to talk about the last experiment. And in this experiment, we actually Marian Schneider in my lab, PhD student, and we basically wanted to see, can high-resolution fMRI contribute to investigations of consciousness? And what we did here, just to wrap up a bit, is to look in phenomenal consciousness, P consciousness from that block. And just that we all at the same page. Consciousness, you find hundreds of definitions, of course. Just a simple one. Consciousness is the state of being awake, thinking, and knowing what is happening around us. I mean, that's obvious, right? The more interesting thing is that for neuroscience, the consciousness problem has been divided into two aspects. One is called the easy problem of consciousness, which I find very hard, by the way, but it's called the easy problem of consciousness, namely to explore the neural correlates of consciousness, so the minimum neural mechanism sufficient for any one specific conscious percept. So that means that if I can say, if this piece in the brain is becoming active, the person will have this and this conscious experience, right? So it's you try to really map the conscious experiences onto neural correlates, onto activity. This can be networks, can be within small things, it can be a neuron. Probably not, but it could be in principle. So that's not defined here. It's only defined that you establish this neural correlate. This neural correlate of consciousness is abbreviated NCC. And the hard problem, which goes back to several people, but mainly to David Chalmers, already in 1995, he says this is this kind of subjective experience that I experience, that I'm aware of, or that I experience how it feels to be a bat and things like that, right? It's this first-person view, which is very, very difficult to approach scientifically. So you quantify somehow the subjective experience of consciousness. I will talk more now about the easy problem, but in a quite challenging way, namely in the way of content-specific neural correlates of consciousness. And again, David Chalmers in 2000 wrote, arguably the most interesting states of consciousness are the states of subjective experience that humans are in at any given moment in time. So it is very related to the hard problem, but we don't say that we can explain that, but we say for these hard problems, these first-person experiences, we find the neural correlate somehow, right? That's the idea. And many people, including Christoph Koch, Marcello Massimi, Chalmers, many people were involved, have set up a research program. How do you find neural content-specific neural correlates of consciousness? And the idea is actually very similar to fMRI studies, where you have a localizer and the main experiment. So what you do is, in a mapping step, you characterize the response properties of a recording site. This can be neurons in electrophysiological animal recordings or intracranial human recordings, but it can also be a voxel or a few voxels in the human brain, right? So this is the mapping step. You first want to know what are these voxels really, really doing. But this is not enough for a neural correlate of consciousness. You need something which works on top of that. And what they come up with, it is one way to do it, is to use ambiguous stimuli, right? This is one way to study consciousness. Because the most important aspect is that you expose a subject now of a multi-stable figure or binocular rivalry where you have two different interpretations. And it's then not the physics which determines what you consciously perceive, but it's your own brain which decides to see this or this one has this content in his mind or that content in his mind. And the point is, can we do this to a level of content? So can we say, this is now the content in the mind of a person, and now it's a different content in the mind of the person. And this we want to do with ultra-high field MRI. One problem is that at least in 2000, there was not much high field MRI. David Chalmers actually was very negative about fMRI to use for that. He praises animal electrophysiological research, but sees also the limits of consciousness and reports in the animal. But he very clearly says that we can trace the difference to the fact that neuron-level studies can monitor representative content in neural systems, whereas imaging studies cannot. So therefore, he was very negative. And I think this has been changed over the last 20 years. So just to show you what we do to really follow exactly this approach, how to map consciousness, I show you now here a stimulus, horizontal and vertical mapping motion. So we go back to MT, and now we map in MT either horizontal or vertical motion by using physical stimuli. So this is just the control, the mapping. And then we get for horizontal motion our columns, which code horizontal motion in MT, which we have mapped already previously, but we do it again for this study in new subjects. And we map vertical motion and columns responding to vertical motion, right? So we have now, if we have high resolution, two distinct representations for this. And now comes the main stimulus. And you have to look at this because I will ask you what you see. So here's a stimulus. So what do you see? Do you see motion? Horizontal motion? Yes. Is someone seeing vertical motion? Yes, some see vertical motion. So if you look in the middle for some time, this will actually switch in your brain. And your brain will decide, although the stimulus has not changed, you will sometimes see horizontal motion, and then your brain suddenly decides, no, it's vertical motion. So your consciousness massively changes. Your consciousness changes, but not physics at all. That's important. Actually, there is no motion. There are just two squares shown left, upper, right, lower, and then two squares shown left, lower, and right, upper. There are two static frames. Everything else is constructed by your brain. And so this is the kind of stimulus to use. And since the horizontal vertical experience flips about 5 to 10 seconds, is ideally suited for fMRI. It's not like a Necker cube, which flips very fast, right? So and what do we see? Actually, we find exactly what we had hoped for, namely that during physical motion, we got this, you see this red-green colored blobs on the left part. We got the horizontal vertical motion columns. On the right, you see the ambiguous motion, where we get the same activity, but it varies, which you see in a moment. And we nicely mapped that these columns were really nicely on the right side, columnar organized. So that's all good for the basic effect. But the main finding is shown here. We indeed, depending on the percept of the subject, if the subject perceives horizontal motion, then the horizontal columns in MT, without a change in the stimulus, boost up and go up in activity. and the vertical go down. If you then see vertical motion, the vertical columns go up and the horizontal go down. And just to show you that, I have a little animation here at the end, which just shows you how this looks like. You see here, zoom in and MT, and red is horizontal and green vertical motion. Now the subject perceives horizontal one, so the reds go up, and now the subject sees vertical motion, the reds go down and the green go up. You see, you get a correlate of the content of consciousness at the level of columnar structures in a tiny area MT. So, and that's what I think allows us now to study consciousness in the human brain. And since my time is almost up, I just want to focus here on the last point. Chalmers' imaging caveat no longer holds. Content-specific mesoscopic neural correlates of consciousness studies are now possible thanks to the technical advancements of ultra-high magnetic field MRI physics and data analysis strategies, as we have described the other speakers in their talks this morning. And finally, I want to say, I will not read this here, just this is for you. I just want to stress that the impact of high-field MRI, not only to solve basic problems, but to really go beyond and shake a bit what we can do to understand cognition, representations in the mind, is opened by this field, and I'm very excited to see where this field is going. Thank you very much.